All right, welcome. Today we're going to get right into some of the most powerful tools in molecular biology, how we build DNA libraries and then how we actually read them. These techniques are, no exaggeration, the bedrock for how we isolate, store, and understand the genetic code of pretty much any living thing.
So here's the game plan. We'll start with the basics. What even is a DNA library? Then we'll look at the two major flavors, genomic and cDNA libraries. After that, we dive into the classic Sanger sequencing method, see how it compares to the newer next-gen stuff, and finally, we'll get practical and learn how to actually interpret the results. Let's get to it!
Okay, first up, what are we even talking about when we say DNA library? Well, before you can study a gene, figure out what it does, how it's controlled, you have to find it and pull it out from the rest of the genome. And that, right there, is where these libraries come into play.
The best way to think about a DNA library is like a literal library for genes. Imagine taking an organism's entire genetic encyclopedia, chopping it up into individual pages or paragraphs, and then storing each little piece inside a bacterial cell. Each bacterium then becomes a living storage unit for one specific chunk of DNA, ready for us to pull out and read whenever we want.
Now, not all libraries are created equal. For one to be actually useful, you need a few things. It's got to have complete coverage, no missing pages from that encyclopedia. You also want a random distribution of all the DNA fragments, so you're not accidentally missing certain parts of the genome. And crucially, you need high representation, which basically just means the odds are very, very good that the specific gene you're hunting for is actually in there.
All right, now we know what a library is, but there are two very different ways to build one. This is the classic showdown, genomic versus cDNA libraries. And which one you choose completely changes the kind of information you're going to get.
So building a genomic library is kind of like a brute force approach. You take the entire genome from an organism, you use what are basically molecular scissors, restriction enzymes, to chop it all up into random pieces. Then you stick each of those little pieces into a vector, put them into bacteria, and you're done. Each bacterial colony now holds a different random piece of the total genetic puzzle.
The huge advantage here, you get everything, the whole shebang. Not just the coding parts of a gene, the exons, but also the introns, the promoters, all the regulatory stuff that tells the gene when to turn on and off. But the downside is just scale. These libraries are enormous. Finding your gene of interest can be a massive headache, a real needle in a haystack situation.
A cDNA library, on the other hand, is a much more targeted strategy. Instead of starting with DNA, you start with messenger RNA from a specific tissue. You then use a special enzyme, reverse transcriptase, to make a DNA copy of that mRNA. So what you're really doing is creating a library of only the genes that are actively being used, the ones being expressed, in that tissue at that exact moment. It's like a genetic snapshot.
The upside here is obvious. Your library is way smaller and much easier to search through because it only contains the protein coding sequences, no introns to worry about. But there's a catch. You get zero information about the gene's promoter or any of its regulatory elements. And if a gene isn't being expressed very much in your tissue sample, it might barely show up in your library or not at all.
So here's the bottom line. It all comes down to your question. If you want to know how a gene is regulated, you absolutely need a genomic library to see its promoter. But if your goal is to, say, make a human protein in bacteria, you want a cDNA library. It gives you just the coding sequence, and you don't have to worry about the introns, which bacteria can't process anyway. Right tool for the right job.
Okay, so you've built your library, you've found the clone with your gene in it. Now what? How do you actually read the sequence of A's, T's, C's, and G's? Well, that brings us to sequencing. And we have to start with the OG, the Nobel-winning method from Frederick Sanger.
The absolute genius of Sanger's method is this little trick involving dideoxyribonucleotides, or DDNTPs. You see, for DNA polymerase to add the next base, it needs a specific chemical hook, a 3' hydroxyl group. These special DDNTPs are missing that hook. So whenever the polymerase accidentally grabs one and adds it to the growing DNA chain, that's it. Full stop, the chain is terminated.
So let's look at the recipe. In your reaction tube, you've got your DNA template, a primer to show the polymerase where to start, and the polymerase enzyme itself. Then you add the building blocks, tons of normal nucleotides, the DNTPs, and just a small strategic amount of those chain-terminating DDNTPs, each one tagged with a different colored fluorescent dye. And here's how the magic happens. The reaction creates millions and millions of DNA fragments, all different lengths, because termination happened at every possible position.
Then you sort all those fragments by size, from shortest to longest. Finally, as each fragment zips past the detector, a laser makes its fluorescent tag glow, and the machine reads the color. The sequence of colors it reads tells you the DNA sequence, one base at a time.
Sanger sequencing absolutely changed the game, and for a long time, it was the only way to do things. It's still incredibly accurate. But technology marches on, and things got a whole lot bigger and a whole lot faster. The difference really comes down to one word: scale. Sanger is like carefully reading one book, word for word. It's precise. Next-generation sequencing, or NGS, is like digitizing the entire Library of Congress in an afternoon. It works by running millions or even billions of sequencing reactions all at once, in parallel. The sheer throughput is just mind-boggling.
So again, it's about the right tool for the job. If you need to confirm one specific mutation in one gene for a patient, Sanger is your gold standard for accuracy. But if you're trying to find brand new genes linked to a disease by sequencing the entire genomes of thousands of people, well, there's just no way you could do that without NGS.
All right, the machine has done its job, and it spits out some data. For Sanger sequencing, that data comes in the form of a chromatogram, and knowing how to read one of these and how to spot when things go wrong is an absolutely essential skill. This right here is the dream. This is what you want to see. Each colored peak is a different base. See how they're all sharp, tall, and perfectly separated? The baseline is flat, no random noise. This is beautiful, high-quality data. You can be very confident in this sequence. Uh-oh. Now what's happening here? You've got two different colored peaks stacked right on top of each other. This means the machine is seeing two different bases at the exact same position. Now this could be bad news, like your sample is contaminated. Or it could be really interesting news, telling you that this individual is heterozygous for that gene, meaning they have two different versions or alleles. And this, well, this is just a mess. The baseline is all over the place, there's tons of little garbage peaks. This is a huge red flag that something went wrong way before the sequencing machine. Usually it means your starting DNA was low quality, impure, or maybe you just didn't use enough. Time to go back to the bench and start over.
So really, you have to think of yourself as a data detective. The chromatogram isn't just giving you a sequence, it's telling you a story about how well your experiment worked from the very first step. Learning to read these traces and troubleshoot them is what separates the pros from the amateurs.
So let's wrap up with this. Think about how insanely fast the COVID-19 vaccines were developed. That was only possible because scientists used next-generation sequencing to read the virus's entire genome in just a few days. That let them pinpoint the spike protein as the target. It's a powerful reminder that these techniques we've been talking about aren't just for textbooks. They are the tools we use to fight pandemics, to develop personalized medicines, and to decode the fundamental instructions for life itself.